It's a pleasure to be able to talk to you a little bit about the work that we've been doing here and show some examples of this, of some technology and some ideas about measuring things outside the laboratory or inside the laboratory, even if you wanted to. But so I called it, when I was asked for the title, I said measuring circadian hormones during sleep, ambulatory profiles in the home. But then I thought, well, actually, maybe more accurately, we should let me see. We should put it more like this. So measuring in bracket circadian hormones, because it's not just circadian ones that we can measure, and the hormones that we might be measuring circadian hormones, but not in a circadian way. So the technology is broad and we can measure hormones during sleep, and the profiles can be done at home. So that's a concept I want to talk about this possibility of high dimensional and high resolution sampling that we have been trying out and proving in healthy volunteers at the moment, but also potentially in patients in the future. So the idea is really to think about this in the concept in this way. So traditionally, if we wanted to really get detailed information about someone, particularly looking at some physiological process that might be moving dynamically or quickly, we have to bring them into the laboratory. And that's the gold standard measurement for things. We take blood samples and really impose a controlled environment on the person, which is quite resource intensive, and it allows you to answer particular questions. But most of the time, particularly from a clinical situation, the person that you're seeing in front of you comes from the real world where there's all these other influences. And it may also be interesting to see what is happening to these rhythmic processes that you can define really well in the lab when they are put in the context of real life. And I'm just Googling this picture of someone sleeping at home, and it always sort of interests me that the pictures of people in these photos always seem to be able to be fast asleep in really bright rooms, which I'm just not sure. It's like that in the movies as well. But anyway, I suppose you wouldn't be able to see them if it was properly dark. But so this is a slide that I quite like that just tries to put the idea of rhythmic hormones in context of the whole body. And so we know that the most potent time of day information is conveyed through the SCN nucleus here through the eye. And so light is this very potent synchronizer, if you like, of the external world. And then we have this clock nucleus here that's the central pacemaker. And it was traditionally assumed that the SCN was the only tissue capable of generating these self-sustaining oscillations, and therefore the only source of daily timing information in mammals. However, it's been proven that molecular clocks are actually present everywhere. So we've got these clocks all around the body in different tissues, and that they're really there, rather than having a master-slave relationship, we've got this more dynamic relationship of synchronization between periphery and the central clocks, and between peripheral clocks, these networks. And then not only is light important, but things like the timing and eating of food become important in the individual's clock milieu, if you like. So, and that's one of the concepts about how disease may occur in people, is that we actually have a mismatch between these central timing cues that are normally synchronized by light and what's happening in the periphery. And you can see how if you've got a clock signal telling you day or night here, but you're eating it at a biological nighttime, you might be upsetting these peripheral clock networks. And then the interest in this has really got much more significant over the last few years. We know that there was a Nobel Prize for literature not literature, Nobel Prize was awarded for the people that discovered the clock genes. And if you read in the newspaper, there's increasingly things about how shift work and disturbed sleep can make you unwell, and hormones that might be linked to particular conditions. A lot of news about daylight saving time shifting and how that could be hurting health. And then perhaps there's some understanding about these hormones like melatonin related to sleep. And then this is from the NHS website that says if you take melatonin, it adds to your body's natural supply, so you fall asleep more quickly. So there's plenty of awareness and interest in this idea of rhythms and the potential of the fact that we could be disturbing them. So one of the things that I'm interested in is like, can we study the nighttime and have a look at what's actually happening a little bit more there in detail in real life to go beyond these associations that we've found or are described about people who are awake at night and how it contributes to health and how that might actually be happening. So the melatonin is the hormone that has really been named the hormone of the biological night, and it's a key pacemaker hormone, if you like, that is used by chronobiologists and others to define someone's circadian phase. And this is a nice illustration from a review paper that shows the parameters of melatonin and what the shape of a curve might look like in a typical kind of person, with low levels during the day, beginning to rise, anticipating the onset of sleep, reaching a midpoint in the middle of the night, and then dropping and coming off, falling away to very low levels during the daytime. And this is a central timing cue that gets examined under dim light in people in order to work out what their natural circadian phase should be. Now, that's a really important hormone that actually peaks in the middle of the night, but usually is unmeasurable in most people, because in order to get to the middle of the night time there, you'd have to wake them up to measure that. We can see that there are plenty of other dynamic things happening overnight as well. And here we've got an example of the cortisol profile. And this is a profile of a healthy person where cortisol has been measured every 10 minutes in a blood sample. And you can see, rather than just being a smooth curve, we've got considerable pulses of cortisol being secreted. And you can see actually the activity of cortisol is very dynamic at nighttime. In this individual, we're beginning to see cortisol pulsing beginning from 2 o'clock in the morning and becoming increasingly high in amplitude, so that by the time they wake up, we've actually had the largest surge of cortisol, and that person is already finished, and it gradually diminishes off during the day. And likewise, if we have a look at this third example down here, another important hormone called aldosterone that helps to regulate blood pressure and fluid volume in people. And we can see that that can be significantly affected by the time that someone's asleep. So in the top figure here, where I'm showing the mouse, and the top figure on the left, where it says aldosterone here, we can see that when someone is normally asleep, we've got pulses of aldosterone and then falling away during the wake time. And if we move the sleep period, we move what's happening with the hormones. And I think all three of these examples really provide evidence for the fact that the time of the day that you measure the hormone and the context of whether you're asleep or awake can make an enormous difference to the levels that you measure, and then therefore the interpretation. So sampling at night is important because there's a lot happening. We can see that we've got peaks of melatonin, we've got peaks and troughs of cortisol, for example, and aldosterone pulsatility can be highest in the nighttime. But in order to sample at night, it has been traditionally quite difficult to do. So here are some pros and cons of different ways that we could measure hormones over the night. So the so-called gold standard of blood allows you to do high resolution measurement. It gives you an idea of what the circulating levels of that hormone might be, and you can certainly control the conditions in which it's measured. But on the other hand, it's pretty invasive because you have to put in a cannula and take blood samples. It's technically difficult, and in some studies even exclude some populations because you just can't reliably keep sampling blood from that person for a whole 24 hours. And it's expensive to admit someone to the hospital. And the other thing that can be, that is useful to point out is that the conditions in which you sample in the lab are contrived. So if the question that you're asking is to what degree that hormone, the changes in the hormone might be relevant to an ambulant population, the laboratory derived high resolution sampling may not be particularly representative, because if you read many of the studies that blood sampling gets done from, for example, they're often young people and small numbers of people, small numbers as well. And likewise, you can only deploy high resolution blood sampling in small numbers of people. You can't scale it up. It's just not practical. Other ways that people can think about looking at what's happening to hormones at night is by taking urine, and that has the advantage of being non-invasive in that you don't have to take any blood or sampling. But And it allows you to measure the metabolites, and it's portable in that someone can carry on normal activities, but it does give limited dynamic insight because you can only at best measure what's happening every time someone goes to the toilet, and generally the whole collection over 24 hours is measured, or a day-night comparison. So its subtleties are lost, and patients don't really like it. You have to carry, I don't know if anyone's ever seen what you actually have to do, but you have to carry a 2-liter container around with you and pee into it every time you need to go to the toilet, and there's a risk of failed collections and so on. A third way that is becoming increasingly popular is measurement in saliva, and that also has the advantage of being non-invasive because you can just chew a cotton swab. It's mass deployable in that you can send many of these out to lots of people and get measurements, and it measures free hormone rather than being a circulating blood sample. So there's the hormone that's present in saliva. But the key disadvantage from my point of view, if we're thinking about sampling at night, is it can't be done during sleep. And as soon as you wake someone up in order to get a sample, you're already disturbing that sleep process. Therefore, you end up with limited time points, which you then have to extrapolate or guess what might be happening before or after your sampling. And it's also prone to user issues. People might forget to do it. They might do something that confounds the result, or do it at the wrong time. And this is just an example to reinforce the fact that individuals vary considerably in terms of what you might measure, and what people have tried to do with these non-invasive measurements before. So here we've got a study in which actually the people were kept in a laboratory and provided hourly saliva samples for 24 hours, which would be an onerous task. But you can see here are 6 individuals, and just a large inter-individual variability in the saliva melatonin profile. And I've just highlighted the axes here to show the huge dynamic range and concentration. So this first person here had a very, very low amount of melatonin being secreted, person A, with a maximum of 4 picogram per mil secreted, and then by contrast we've got a range up to 80 on this lower figure E and F. So one of the challenge that I thought about was how, or we have been thinking about in our lab, is how could we assess the dynamic biological change at night, but to do it outside the laboratory, to be able to capture the sorts of dynamic changes that occur in the middle of the night when people are asleep, but without having to take blood samples or put someone in the laboratory. And then the second point was, could we contextualize the data to the participants' real world experience? So could we say, rather than enforcing a sleep schedule on people like they would have to have in the lab, how does that hormone profile or that change in those changes in hormones relate to what that person actually does day to day or at that time that they were sampled? And the solution that was chosen was to use a technique called microdialysis. And what microdialysis has been around for quite a long time, for decades, and it's basically a synthetic capillary that is made up of a semi-permeable membrane that you place in tissue. And here is a cartoon illustration of the kind of ones that we use, which get placed subcutaneously, so just below the surface of the skin. They're about 3 centimeters long and about 1 millimeter thick, so very small. And they have a semi-permeable membrane that, when they get placed in the tissue, allows hormones and metabolites to travel across in a concentration gradient and to give you a readout of what's happening at the tissue at any particular time point. And what's useful about interstitial fluid? Well, this table gives us some idea about why it's a good sampling medium. And here we can see this top row compares interstitial fluid against other types of body fluids that you might sample from. And so you can see there's a wide range of molecular weight molecules that can be present in interstitial fluid. And this is up to 1 megadalton, and that's just for reference. If we think about something like cortisol or melatonin, the molecular weight of those molecules is less than is less than 1 kilodalton, about 300 or 400 daltons. The lag time between blood and the interstitial fluid can be quite short. It doesn't require stimulation, and there's a lot of evidence for the presence of molecules in the interstitial fluid. And if we think about the most commonly sampled interstitial fluid example would be glucose, and if the interstitial fluid glucose monitors that are used in type 1 diabetes sample exactly an interstitial fluid. And then if we have a look again here, we can see the chemical composition has quite a number of similarities to plasma, and particularly in terms of its electrolytes. And is a rich source really for us to measure lots of different types of hormones and metabolites. So how do we obtain these dynamic profiles? So we said we wanted to be able to measure these hormones over time and to capture them without blood, and we have the solution of the interstitial microdialysis, which is being used. So how does that work in practice? Well, what we do is we implant the membrane superficially below the skin, so just a millimeter or two below the skin of the abdomen. It doesn't require local anesthetic to do, and it's not painful once the membrane is in. Then we push very slowly across that membrane some isotonic saline, so basically salty water that is the same osmolality as the surrounding tissue, and then that allows for hormones or metabolites that are around the probe to diffuse across the probe membrane into the fluid, which can then exits the end of the probe and as microdialysate and into our sample collector. And that's what the sample collector looks like, and that is worn around the waist. So then this is a wearable system that allows us to continuously sample the interstitial fluid for more than 24, 48, 72 hours at a time, and obtain samples every 20 minutes of a number of hormones and metabolites. It's very flexible. It can be used from the neonates to old age. We've had neonates who were sampled in the hospital here in Bristol undergoing cardiac surgery, had the procedure done, who would otherwise be impossible to measure these things because of the volume of blood that would be required. It could be used in the laboratory, but the real benefit of this is that once this is set up and the person is wearing it like this in the cartoon here on the top right, they can just go home and carry on almost all normal activities. And this is the kind of data that we can obtain from that. So here we've measured examples of 3 hormones that we profiled in more than 200 healthy volunteers in a paper that we just published recently. And you can see the features of these adrenal steroids that become immediately apparent here. So we have evidence for a daily rhythm of cortisol in each of these lighter blue lines at individual profiles. And then the darker line shows the rolling mean and centiles. So you can immediately get a sense of the inter-individual variation of particular hormones, with that being highest probably for aldosterone more than cortisol. And here we have the mean sleep period of the cohort, where we can see that low hormone concentrations occur just after the onset of sleep on average and peak just as a person is waking up. Aldosterone is a much more pulsatile hormone that has a lot more individual variability, but nevertheless still has evidence for a daily rhythm. And we did confirm that by applying a multiple component cosinus model that confirmed that all of these hormones had both evidence for a daily fit, but also for an 8-hour period as well. And that just made me want to move on to the point where we consider the difference between applying mean or median data from a population to what's actually happening in the individual, and the possibility that we now, with this kind of measurement technology where we can measure multiple hormones at the same time, we can really start to look at individual profiles and as opposed to considering what would be happening on average to that person. Now this is a complicated figure which shows mean data, and I'll just take a moment to explain that. So here, this is a circular heat map of 7 different hormones that we measured in microdialysate. And each of these cells represents a 20-minute time point over the 24-hour day. And this data has been Z-scored. So the highest concentrations are in these dark red colors and the lower concentrations in blue. And then we have the mean sleep onset and offset for the whole cohort around the rim here. And this immediately allows us to look at patterns of the hormones and to see how that daily rhythm applies to the average of the data. So we can see that the nadir time of all these hormones is occurring after the person has gone to sleep, but at slightly different times. And then really importantly, that these hormones are starting to rise early in the morning before someone is even awake, and peaking right around the end of the sleep offset time, with some variation between the hormones. Now, if you look at that, you can see that, at any given time point, each of these hormones is likely to be correlated with each other. But on an individual basis, that is quite often what we saw in our data. So here is an example of a profile collected of cortisol and aldosterone in the same person over the 24-hour periods. And we can see in this case that those hormones were highly correlated with each other. In fact, they were almost indistinguishable in terms of their dynamic properties, with the peak occurring clearly at the end of the sleep period and nadirs overnight. But then again, on the other extreme, at the other end of the correlation comparison continuum, we could see profiles like this, in which where the hormones were clearly not correlated with each other statistically, and we had quite pulsatile aldosterone that was occurring without equivalent pulses of cortisol across the same time. But then again, we have this concurrent peak of both hormones right at the end of the sleep period that's preserved. So this kind of technology allows us to observe different mean and population trends, so how things might have daily rhythms, but then to observe for a particular individual how the relationships between hormones might differ from what we see in the mean, might actually be quite distinct. And then in order to really think about that data in a more quantitative way, what we did was to extract some dynamic markers out of these profiles. So here we can see in this red line is one of the healthy volunteers that we sampled in our project, with a background mean of 214 healthy people. And then we could use that profile to extract features out of there. And critically, many of these features are features that are defined by sampling periods that occur overnight or during the sleep period. So we can define area under the curve at different time windows. We can look and find the evening nadir for those, the time that the hormones start being secreted, the time that they peak. And using that, we can stratify profiles and quantify the variability of normality, and then apply those normal distributions when we're looking at profiles that we might suspect are abnormal. And here is the red profile on the left, shown as in relation to the distribution of area under the curve at different times of the day. And you can see that it looks like a normal profile, and we can quantify that to be normal using the area under the curve variable here. But then if we can take an abnormal profile, and this is what we call pituitary Cushing. So this is a rare endocrine disease where the pituitary gland is over-secreting a hormone that stimulates the adrenal gland to produce excessive amounts of cortisol. And we can see that profile looks normal visually, but now we can quantify that mathematically and really define that indeed the highest level of abnormality is actually occurring in the evening and at nighttime. And I've just highlighted the sleep period of this pituitary Cushing's person. I think this really illustrates that a great deal of the abnormality is actually occurring at night. And if we'd taken a traditional 9 a.m. blood sample on this person, we might have seen that fell within the normal range. And likewise, on the right here, we've got another disease called primary aldosteronism, which is related to high blood pressure. we can see how this visually abnormal profile can be quantified using these dynamic markers to show that it's between 9 p.m. and 9 a.m. that the most deviation from the healthy normal distribution occurs. And then again, if we can see that by sampling during the sleep period, we've gathered a great deal more information about this profile and the abnormality of it than if we had just sampled during the day, or and especially more than if we'd just taken one time point. One of the things I was particularly interested in as part of my PhD is the technology for measuring cortisol in the interstitial fluid had been proven at that point, but I was keen to see whether we could add melatonin onto that, and that way we could have the circadian pacemakers, or the marker of the biological night, measured along at the same time as cortisol, and working together with Dr. Martin van Vasten and the UCMG in the Netherlands, we developed a method that allowed us to do that in the interstitial fluid. And we can see this is a nice healthy person who was carrying on normal activities at home and sleeping to their regular sleep schedule. And we've been able to recover cortisol here in red and their melatonin profile in black. So this is not a dim light melatonin. This is a natural, or their normal melatonin under regular life conditions. And we can really nicely see how this person's sleep-wake and the melatonin and cortisol rhythm are all aligned together. So we have the onset of melatonin occurring right just before their habitual bedtime, peaking overnight, coming off as they wake up, which is well coordinated with the peak of their cortisol. So it's this kind of example that I think really illustrates how we can potentially start to look at both healthy alignment and then, what would happen if we were to put a perturbation or a disturbance into someone. Would these relationships be maintained? What would potentially start to be unpicked? And as part of my PhD, what we did was to really start to have a look at using the technology at the interstitial fluid measurements, measuring cortisol and melatonin together. How robust was it in the same individual? And we can see here, these data come from the same individual where we sampled them over 4 separate nights, and the red is the cortisol, and we can really, and the blue is melatonin. It's been aligned by the onset of cortisol in the morning, and you can really tell how this person is synchronized. These 2 top profiles here from 1 week, and the lower 2 are from the 2nd week that they were sampled. We gave them a light perturbation close to the time that they went to bed, and you can see that seems to have, there's evidence here that this has caused a melatonin delay here. The dark circles and dark squares are the nights where they had received bright light shortly before, or in the evening before they went to bed. We used a special bright visor that shone light into their eyes here for this. And then if we align the profile, these same profiles by the time that the melatonin offset occurs, so this is when the melatonin has fallen to 25% of its peak level here, we can really, and the yellow here is the time that they had the bright light shining, we can see how the melatonin has potentially been affected by the light here. It was starting to rise and it's been delayed. But then, and we can really also see, I think, how we have the same the phase relationship between cortisol and melatonin here, where we have, when we align it this way, we can really see how the peak of melatonin is occurring right at the nadir of the cortisol time, and then how we have alignment of an inverse relationship with peak of cortisol occurring just as melatonin is reaching its offset period. And now, as I was mentioning, this kind of measurement allows us to study voluntary or involuntary perturbations. And I just wanted to show this example because it was one that we didn't intend to happen. But in this particular project, this healthy volunteer was being sampled for 2 consecutive days. They didn't have any restrictions on when they were allowed to be asleep or awake. And this first night was a self-selected normal sleep night. So this person went to bed at the time that they wanted to go to bed and woke up when they naturally wish to, and we can see that we have this nice anticipatory peak of cortisol that was rising here from about 3 o'clock in the morning, peaking just at the end of the sleep period, with the melatonin really straddling the sleep period, as we might expect. Now, on the second night, we had quite a different looking profile, and you can see here what happened. So there was an unintended sleep delay for this person. So they were kept awake by a loud party. We can see the melatonin began to rise just as it did the previous day. So this was unaffected by the change in their sleep onset. They eventually did go to sleep right at the time when here just at 4:30 in the morning, and if we refer back to the previous day, this was right at the time when the cortisol would normally be moving towards its peak, but as he went back to sleep, his cortisol actually went down and then rose again, peaking to his delayed wake. So you can see this acute sleep change we were able to observe. We didn't intend for this to happen, but it was something that we could see really has significantly changed the cortisol dynamics in that morning. And you could see that if we did a single time point cortisol measure, we would have captured none of that information. And if we didn't know that this sleep perturbation had occurred, we might misinterpret the results entirely. Now, mostly what I've talked about is melatonin just now, and then also the adrenal corticosteroid hormones, particularly cortisol. But one of the next research areas of interest is whether or not we can also measure other aspects of the stress response, and those being the outputs from sympathetic adrenal medullary axis. So in the adrenal cortex is where we would find cortisol and aldosterone, but from the medulla we get our adrenaline and noradrenaline, which are obviously crucial for normal homeostasis, but also the fight and flight hormones that react acutely to stressful situations. And this is pilot data that we've recently received, and I'm really happy to be able to show you this, where we were able to capture both free noradrenaline and adrenaline in a healthy volunteer and really see the dynamic changes that have been occurring with that. And again, the stark day-night differences that we might see. And this noradrenaline daily profile here is consistent with what we observed in laboratory studies in the past, where it's known to have a diurnal rhythm. And it's really lovely to be able to see us replicating this free noradrenaline hormone in someone who's walking around without needing to take any blood samples. The other possibilities that we've begun to investigate, or are active investigating as well, is the potential measurement of other kinds of metabolites. And here's an example of eating rhythms that we can see. And here we've got the amino acid called leucine, as we measured it in both plasma and tissue. And we can see, and these vertical dashed lines are meals. This person was sampled in the laboratory, and the red lines are the plasma, and the teal color are tissue. And we can see how these amino acids are tracking across the day, with the tissue reflecting the plasma really nicely. And what we can observe here, what's happening at night. And this methodology now gives us the option to have a look at the potential effects on these peripheral clocks or peripheral metabolism, for example, on people who might be having an acute shift work pattern, who normally have a normal day-night rhythm, but then suddenly go on a shift and work at night and eat at night, and what might happen to these amino acid rhythms. We can without needing to take blood, we can do these high resolution profiles and see what's happening to some of these peripheral metabolite rhythms. The next, and probably I think the final thing to talk about for today, would be taking this one step further, and that was contextualizing that biological data and seeing if we can take some other measurements using wearable devices and self-reports to contextualize that information outside the laboratory. And this is what I kind of think about it like a 360-degree view, where we have the participant in the middle. We have the opportunity to take real biological sample signals using the rhythm microdialysis sampler to measure an actual hormone, potentially a circadian clock hormone, and then measure things like activity, their sleep, glucose, and other parameters at the same time to really see how this hormonal information can be placed in the context of that person's life. And here is an example of an ambulatory participant where we were able to do that and the kinds of information that you can gather. So here we can see the melatonin and cortisol profile up the top. We can see peripheral skin temperature in the orange on the second here, which really nicely frames the time the person spends in bed. Their glucose rhythm in green, and what's happening with that over the day and night. And then 3 different amino acid metabolites that we can see, leucine, isoleucine, and valine, which are particularly implicated in, for example, the development of type 2 diabetes. And we can see how those hormones track in the context of their circadian clock hormones, their sleep period, and their glucose and body temperature. So it really just gives you a really rich kind of data set, which you can then begin to look at planning further experiments or examining the possibility of relationships between these different processes. Using these kind of visualizations that I showed before with the circular heat map, we can see group and individual differences together. So here we've got 11 participants who were allowed to carry on normal activities while they were being sampled over the 2 to 4 consecutive days each, and we can see their peripheral skin temperature measurements in relation to sleep and wake times, and the nice peripheral vasodilation and heat dumping that occurs with the finger temperature rising overnight here, and how that relates to how that's sort of coordinated in these normally sleeping day-night participants who, in relation to their melatonin, which is also high at the same time as the temperature. And then also we can measure light using the wearable devices and see how these individual light patterns might particularly relate to any one of these rings. So this kind of visualization, I think, is really nice in the way you can see the group trends, but then we can look at the individual ring and see how that particular person might be similar or different to the remainders in the group. And then this is another example. So you saw the noradrenaline hormone before. So this is one of the free catecholamines that's really important for regulating heart rate and the fight and flight response and so on. And we can measure the hormone together, and that gives us a certain amount of information. But then if we contextualize that with wearable data, it really gives us some fascinating insights. So here if I plot that noradrenaline against the person's heart rate, we can see how closely they relate to each other. And again, with that noradrenaline and activity, we can see how important heart rate and sympathetic output really are linked to each other, and how that relates to how we can really see those relationships just by measuring activity and heart rate at the same time. And then we can really layer that up into quite complex levels of data. And this is where the project that I'm working on at the moment is to collect all these sorts of things and put them together in healthy people and people that we know have got sympathetic disorders of increased sympathetic output, where we can capture their melatonin and cortisol, so their circadian pacemakers, so measured obviously in masked conditions. We can see their light exposure and finger and wrist temperature to look at the patterns and the daily rhythms there. We can see blood pressure, ambulatory. We can measure the heart rate. We can measure glucose and other things like dopamine in response to particular types of food. We can look at activity and how they might relate to any of these other particular parameters, and heart rate variability in the hormones themselves. And one of the things, particularly since this is thinking about sleep, is at some stage it would be nice to really look, rather than this grey box of being asleep or awake, is to consider how a particular sleep stage, REM versus non-REM, for example, might be able to unpick some of these oscillations or these more subtle variations that are occurring in some of these hormones at night, to give us even more information about how there may be system-wide interactions between all of these different things. So just to finish off, and I think I'm running pretty much on time, the night is full of dynamics. And so when we go to sleep, we don't only have what we might, most people would know that sleep stages cycle through, but our hormones are extremely dynamic at nighttime. So we've got peaks of melatonin, we've got troughs of cortisol and peaks of cortisol, we've got pulsatile aldosterone, we've got quiescence of the noradrenaline and the sympathetic output rapidly changing as we wake up and get out of bed. Measuring at night without disturbance. So that's what we can do with this microdialysis system. It provides quite a lot of new opportunities, I think, to be able to observe without interference, so that we can see what is happening without waking someone up to do a blood sample or a saliva sample. And I think it has an option to really give extra power to field studies, which previously or mostly rely on indirect or passive sensing, to give that biological insight in addition to those other things that you can get from self-report or passive wearable devices. And then it opens up this other very complicated realm of high dimensional data, where we can potentially begin to contextualize the measurements of any particular things, have a look at an individual experience, and I guess with mathematicians and other people to start sort of building models that might predict how, if you give a perturbation to one system, what something else might react, or to see how, to understand disease physiology, or how somebody might react to particular changes in circumstances. So then my final slide is just to acknowledge, hopefully, most of the important people that have been involved with this work. So we've got the Urhythm device, that's the star of the microdialysis. We've got Dr. Eda Zavala, who's based in the University of Birmingham, who I've worked closely with to bounce off ideas and develop the idea of the dynamic markers and the mathematical approach. Martijn van Vasten in UCMG in the Netherlands, who's developed some of the amazing mass spec methods that have allowed us to measure these hormones at very low concentrations. Deborah Skeen in Surrey, who is the chronobiology person who's really supported and helped me through a lot of this work, and then the Ultradian Consortium, which we developed and did the sampling in the healthy volunteer on those 200 healthy volunteers to see the cortisol and other steroid dynamics. And then, of course, Professor Stafford Lightman, who is here in the University of Bristol. And with that, I will finish and answer any questions. Thank you very much.